I'll hand it over to Pierre Bellac for our next talk in just a moment, and he'll tell you a little bit about himself. Just to kind of reiterate, feel free to ask questions at any time. I'll be monitoring, and I will interrupt with your questions as appropriate. So again, delighted to have Pierre Bellac to tell us about brain parcellation. Take it away, Pierre.
Okay, great. Thanks, Tal. Obviously a pleasure to be virtually here for this Neurohackademy. So just a couple of words on my background, so you have an idea of where I'm coming from. I trained into mathematics and modeling, artificial intelligence, and then I started working with brain imaging during my PhD, mostly on modeling still, but like doing a lot of applications. And for the past 10 years, I've had a lab at Université de Montréal, where I try to apply fMRI to discover biomarkers in Alzheimer's disease, which I proved more challenging than I would have hoped for, but that's what I'm working on. So yeah, I was initially a prof in computer science. Now I'm in psychology. So I basically spend my whole life in between the world of neuroscience and modeling. So today I'm going to be talking about brain parcellation, which is one of the tools I've been relying on again and again since I'm a master's student, basically. Actually, the conclusion of my master thesis was that we needed better parcellation to characterize the graphs of patients recovering from stroke. And unfortunately, 20 years almost has passed, and I'm still stuck working on that darn problem. So let's get started. I'm gonna just share my screen, and the material is on Jupyter Hub if you want to follow along. So here you should be able to see my Jupyter Hub window. Let me make that bigger. And inside curriculum th-parcellation-Bellec, you should see a bunch of files, way too many files, but there's only one notebook. That notebook is called Functional Parcellation Introduction, or intro, whatever. You can open that. Now, what if you don't see it? It's because I've uploaded it very late. And if you let your server run, maybe that folder is not up to date, in which case you may want to click the plus here. That's going to give you this screen there, and you can open a terminal. And inside the terminal, you're going to type this instruction, git puller nh curriculum master curriculum. That's going to give basically update the curriculum. So I'm going to do it. It's already up to date in my case, but maybe it's not going to be in your case. I'm just going to real quick put that in the chat. Actually, there's a chat. There's a chat.
I'll do it. It's fine. You can keep going.
You'll do it. Thanks, Tal. A real pleasure. So I'm going to quit this interface, but you can, what you can do is just like open that notebook and you can follow. Now I'm going to actually switch to a traditional notebook and not Jupyter Lab, because I want to be able to go into slide mode. So my notebook being here at the top. So, because in the regular notebook interface, there's this nifty little extension here that lets me use a slideshow mode. So, and I'm actually even go full screen with that. So now you should see just my slide. Does this work? Tal? Yeah. Wonderful. So, introduction to functional brain parcellation. So, what is a brain parcellation? Well, I use that word very loosely, but the idea is to divide the brain into pieces. The most simple way is like the pieces cover the whole brain and there's no overlap between them. And you're either in a piece or not in a piece. So that's what's represented on that first slide. That's the iconic, classic Yeo-Crinan 7-cluster parcellation right there. And each color codes for a certain parcel of the brain. You can give them a name. We're going to get back to that parcellation in a short while. But that's what it is. Now, why would you want to do that? it's funny because there's kind of this whole tradition of parcellating the brain. So there's an entire history of people doing that in neuroscience, dating back to at least Brodmann, who looked at the characteristic of the laminar organization in the cortex and found that they were kind of like modes of organization, and he proposed to slice a particular brain into a subset of areas. And that was a really popular thing to do, and there was many iterations based on that. But that's for structural aspects. But in terms of function, there's this beautiful review by Francisco Varela from 2001, which I keep citing, that really suggested that there are functional assemblies that emerge during cognitive tasks in a transient manner. And those functional assemblies are shaped at multiple scales of organization. So locally on the cortex, you have a lot of connectivity that's directly in the gray matter through the cytoarchitecture. So those can give rise to like highly specialized, highly homogeneous local assemblies. Probably the finest scale that we have available would be the microcolumn. But then there's also large scale connectivity in the brain, basically regardless of spatial distance. I mean, even with the rest of the body, right? There are axonal projections that go all the way through the spinal cord. So you also have emergence of distributed functional assemblies. And that review covers different types of electrophysiological measures, including intracortical electrophysiology, as well as scalp-based. So even though my talk is going to be with functional magnetic resonance imaging, I think that the type of objects, type of phenomenon we try to characterize with functional parcellation are really those local and distributed functional assemblies. So there's no set terminology, but you'll notice that in my talk, I'm going to talk about regions for those local assemblies, typically parcels which are connected in space, network for assemblies that are distributed at a large scale, and parcel for whichever, interchangeably. So basically, parcellation in that context is really to try to understand the brain and to try to understand the functional pieces of the brain. So it's very much of a neuroscience objective. Now, there's also a more methods objective. You can also think about parcellation just as a way to reduce the dimensionality of your data. So traditional fMRI data is, in the order of 10,000 voxels in the gray matter, something probably like 40,000, 50,000, depending on the size of the head of your subject and the exact resolution you have. With modern sequence, accelerated sequence, you can go down to about 2 millimeter isotropic with still decent signal-to-noise ratio, in which case you're cracking over 100,000 voxels in the gray matter, actually close to 500,000, again, depending on the specifics. So that's huge. So parcellating lets you go back to maybe like 150 regions, 200 regions, that's much more amenable. And for people who do graph-based analysis of the brain, this is very popular. So they're going to use a parcellation such as AAL, that's represented in the left, just compute average activity in those regions and use that to build a graph based on maybe the correlation of the time series. And that small graph on the top left is, you can look at its properties, see if the properties change based on, some behavioral, cognitive measure, whatnot. But obviously, you're losing a lot of details in your data. So you can use other parcellation, which maybe capture better some aspect of brain function. So even at a single resolution, you have many different parcels available to you. Here you have a couple, just mentioning the CSIC parcel, the Cameron Craddock's parcel here with 200 regions or 400 regions, which are pretty popular. People use a lot to build graphs. But then you can also try to scale up and get maybe 1,000 regions, or 3,000 regions. Those would be the graph at the very bottom, where you see a lot of details, a lot of complexities that may be missed if you go with cruder parcellation. So obviously there are implications of using more parcels in your analysis in terms of statistical power, in terms of what properties of the graph you'll be able to capture. So all those are important considerations. And so during that little talk, I'm going to try to touch about important concepts about like how you build parcellation and how you judge that the parcellation is good in a sense.
Hey Pierre, this is just sort of an aside question, just a quick one. Someone's asking what software you use to plot the degrees, well, the nodes and the edges here.
So that's GPHI. Does that make sense? GEPHI. It makes beautiful graphs. It's not necessarily the most modern interface, but that's what I use to do those figures. And I tried to reproduce them using Nylearn for this tutorial, and I wasn't able to do some things that I was satisfied with. So for now, that would be my recommendation.
Sorry, one more just clarification question. Someone's asking if you can elaborate on what you mean by graphs. What do you mean by parcels can be used as nodes to approximate brain networks as graphs?
Of course. So a graph is an object that's basically defined by a set of nodes. And when you use a graph representation of brain networks, the nodes are going to stand for regions, typically, or more generally parcels. And then those nodes can be connected by edges. Those edges can be either binary, you have one or not. They can be weighted, you can give a strength to the edge, or they can be multiplexed. You can imagine having several measures of connectivity between any two given nodes. So this is a sort of popular way of characterizing brain networks by approximating them to the graph. So in those graphs, each little point is a brain parcel. So brain parcels are a key tool to build them. So in terms of the format of the presentation, I'm really used to go through that material as traditional slides, but because this is Neurohackademy, I've decided to change a little bit the format and try to throw in some code here to show you how you can essentially reproduce most of the figures of the presentation using Nylearn and open data. So there's in the folders, there's a file requirements.txt. I mean, there's only a few requirements, basically Nylearn and Matplotlib and maybe Rise for the slides. And we're going to use a few data sets. And by running this cell here, you're going to get a local cache of the data sets, but it should be already synced on the server. So I think should not do those messages you see under if you rerun it. So a couple of parcellation, the BAS parcellation, the YO parcellation, we're going to get back to what they are. And then a couple of subjects from the ADHD 200 samples that have already been preprocessed using the Athena pipeline. So we're going to try to create parcels from those data together. So before we get started with the parcellation, real quick, we're going to be doing functional MRI. And I want to emphasize that functional MRI is not neuronal activity. You know, I was talking about electrophysiology and the VIALA review, but what we have in this tutorial are measures of blood oxygenation over time. And, you know, I don't know to what degree you're familiar with that, but this is a key sort of limitation, I guess, at least in terms of interpretation of what we do, that we're not directly dealing with neuronal measures here. Now, there is a beautiful coupling between neuronal activity and blood oxygenation locally in the brain. And this is because the blood supply to the brain starts with huge highways and breaks into small roads and end up into those tiny microcapillaries that are directly next to the neuron. And when the neuron works, they need fuel, and the capillaries respond by becoming bigger and getting more fuel, which is oxygenated blood, coming in. And it just so happened that oxygenated blood and deoxygenated blood, or the hemoglobin part in the blood, they have different magnetic property. So that's going to mess up with your magnetic field, and you can make an image of it with a very fine spatial resolution. And by that, I mean, you know, in the order of the millimeter isotropic. So I'm not going to, comment more on that. I've linked a little intro to fMRI. I've given a few weeks back. If you want to check that out, there's tons of really excellent resources on that online. But if you're doing fMRI research in parcellation, you absolutely need to understand what kind of signal you're manipulating, because it is a very complex signal that is not easy to interpret. Now, concretely, it's actually the origin of signal is complex, but what the signal looks like is simple. It's a 4D data set. So you've got a series of brain volumes that are being acquired. So here's a beautiful graph from the Nylon documentation. You have the different time samples here from T all the way from T-3. And at each sample, you're acquiring a full brain image, which can take as little as 500 milliseconds using multi-slice accelerated sequences. So, it's pretty amazing that we're able to do that. And typically what we do is that we're going to get those series of brain data and convert them into a simple array. And I know that Elizabeth Dupré covered that already in her tutorial, so I'm not going to go too long about this. But basically, when you have a parcellation, what you want to do is simply, you have a series of little regions. What you want to do is your array, each column is going to be a region, and you want to average the temporal activity in that region. And that's going to give you a column in your matrix. So if you want a brain parcel, here we are importing the plotting tools from Nylon. And we have this very handy little tool that's called plot arrow I standing for region of interest. But it works for any parcellation. It doesn't have to be connected necessarily spatially. And we're going to tell it to plot a particular parcellation. So at the beginning, I fetched the Basque parcellation, and I called the fetched object Basque. And Basque comes with different flavor. We're going to talk about that in a little while. Here, I'm taking the scale 444 flavor, which has 444 regions. I'm just providing a title. I'm putting on the color bar. I'm using a jet-like color scale just to make jet haters rage a bit. Yeah, there's a lot of controversy about this color bar, but it's good to differentiate lots of region. So I'm using it for this.
I'm sorry, Pierre, I have to stop you. We had
yes.
a very clear no jet policy, so we're going to shut this down now. And thank you all for. Sorry.
I broke the policy. I am absolutely sorry. Let me show you a very cute dog to try to.
Oh, that's okay. Everything's okay now. Right. Proceed. Okay. Sorry.
Great. I'm going to try not to do it again. I think I have very little jet in this presentation after that. So here you can see those different parcels. I mean, I've already shown you that. So here's a repeat, but with the code to do it. And then we're going to get the regions. And for that, as Elizabeth explained, we have a zoo of different maskers. And to work with those type of parcels, which are, just little region characterized by just a number, that's called a labels masker. So we're going to use the nifty labels masker here. And we're going to provide the name of the parcellation we want to use. And then a bunch of little options for how to pre-process our time series. So of note, we're going to do a bit of detrending, removing a slow time drift, which is really like the one step that you should always do in your pre-processing, because those are a major source of noise and will completely destroy the structure of your signal if you don't do it. We're going to standardize the data. That means that all our time series are now going to have a zero mean and unit variance. We're going to smooth spatially the data a little bit, and then some boring options. And we apply fit, which is we initialize the masker. And we don't really need to specify any data to initialize it because we've already provided the mask. So there's no parameter to the fit part. And then we're going to transform a particular data set, the first one in our collection. And we're getting back some time series right here. And so time series is an NumPy array, and it's going to have a shape of 176 times 444. And why? It's because there's 444 parcels in that particular parcellation, and there's 176 time points. So that array is the array I was showing you earlier with like the little brain on top and all those columns. So let's plot one of the columns to see what it looks like. We're going to select the parcel number 316, And why 316 is because I went and visualized the parcels, and I've noticed that 316 is right in the posterior cingulate cortex. And the PCC is a very interesting region. We're going to talk a little bit about why it's interesting. But so I decided to just look at what was going on there. And you can see all those like spikes, those crazy spikes, and they're slow. Right here, we are very low. We are probably second 35, something like that. And then there's a spike coming at second 50, or time point 50, sorry, and going back down probably at time point 60 or something like that. So there's probably roughly, 20 time points to have this big spike coming. And there's a few seconds to get one time point. So roughly those spikes, you're going to see them take about 30 seconds to a minute to come and go. And that is a very common phenomenon, the slow fluctuation in spontaneous activity in the bold signal. So that's very intriguing. What are they coming from? And one of the key experiments that was made by Bisval a while back was to try to correlate this kind of activity with the activity of the rest of the voxels in the brain. And he found that there was all those hotspots of high correlation in the brain, and that those correlations were not distributed randomly at all, but seems to follow well-known, or at least for some of them, network of brain regions that are involved in similar cognitive operations. The simpler being the sensory motor network, which is very well understood. And if you put a seed in pretty much any region of sensory motor network, you find the entire network, which is neat. Here we use the PCC, and what we get is like, a big blob around the PCC and the medial frontal cortex, and those like two blobs laterally, which are temporal parietal juncture, TPJ. And that's the default mode network. So that's a classic way of finding the default mode network. That's how, oh my God, Mike Gracious found it in his 2003 PNS paper. So can we do that with our parcel and our randomly selected subject? I hope so.
Pierre, a couple of quick questions.
Yeah.
One, I noticed both you and Elizabeth are using some filtering and smoothing within the NiLearn masker object. Is this in addition to any of those similar steps that happen during pre-processing of data?
That is a very good point. And I suspect those data were actually already fully pre-processed and that I could have just done nothing. So, and in general, it's a bad idea to double pre-process your time series. Unfortunately, here, I'm not 100% sure, and in depth, I just did it.
And the other question, can you comment a bit about differences, the different NiLearn masking options? Actually, I'm going to suggest that maybe Pierre doesn't answer that because Elizabeth, I think, covered that. So you can go back and watch the NiLearn lecture. And also, there's a really nice, I'll paste the URL. There's a really nice example of that in the NiLearn documentation.
Absolutely. NiLearn has a comprehensive doc, and Elizabeth covered those topics much more extensively than I'm going to be able to do today because I need to cover the more stuff on the parcellation part. So, but here we're going to use another masker. So maybe that's going to answer partly your question. And this other masker is going to give us the voxel level data. So in the previous masker, we got 444 time series because we extracted averages on those parcels. And here, it's going to compute a mask of the brain. So when we fit here, we need to specify a data set to compute the mask. So that's the difference with the previous masker. We have similar options here. And then we're transforming that data set. But instead of having 444 regions, now we have 69,681 voxels. And we're going to be using that to create a seed-based connectivity map, just like I showed you in my slide. And obviously, there's better, more efficient ways to do that in Python. But here, I'm showing you a bit of the math, so you get an idea of what it is. Basically, a correlation is essentially just a dot product. for time series that have been corrected to zero mid and unit variance. And the thing is, they already are like that, because if you remember the masker, we use the standardized option at true. So if we wanted to compute a correlation, essentially all we have to do is use NumPy. So I'm importing NumPy here, using the dot product, elements by element here. Here we go. There's just a normalization by the number of type point we've got. And that's going to give me a correlation map. Now I got here in that vector, I got a series of value for each voxel, but it's not really a brain map anymore. So to reshape it as a brain map, I use masker and a method that's called inverse transform that takes me data that were transformed as an array and put it back as a brain map. And then I can look at this map using this plotting tool called plot stat map here. And there's a few options. I'm using cut coordinates that are identical to that slide I presented you at the beginning, which I've done years and years ago, by the way. So I was pretty happy. It reproduces pretty well on a single subject here, randomly selected with neither. Same threshold actually as in the previous slide. I'm showing only voxel with a correlation above 0.5. And as you can see, there's pretty high correlation both in the PCC and the frontal medial cortex. And you have those two little regions in the TPJ. So yeah, default mode network, pretty much every time it works. It can be more or less clean, more or less noisy, but that's a very robust finding even at an individual level. But now, can we use that to define the network in its entirety? Here we just have a voxel-based map, but what we really want is to be able to segment that network. And to do
clarification,
that,
just real quick clarification question: does that threshold account for multiple comparisons?
No, not at all. It's because 0.5 is big. It's not a significance threshold; it's a substantiality threshold. But if you were looking at significance with the type of degree of freedom we have, I would say probably around 0.3 would be our threshold. So yeah, to be able to take our finding with a seed-based and use that to build a network, I'm going to take a little detour and tell you about what a connectome is. So a connectome, I'm going to use a parcellation once again. So here you have one on the left. It's not actually the 444 parcels. This one has more like 1200, but same idea. And for each region, we've already seen we can have time series. So we can compute this correlation matrix that is region times region. So 1200 times 1200. And for each point in my matrix, what I have is a correlation of the activity of a given pair of region. All right. And one way of thinking about what that matrix tells us is that each column is a seed-based connectivity map. Why? It's because basic linear algebra, here I have column J, with J standing for one of my parcels, one brain region. And here I have all the values I, with the index of the rows, which tell me the connectivity of that region J with all the other region I's in the brain. So if I take those values and those regions, and each region I color based on the value of the correlation, what I have is a seed-based connectivity map. It's not an analogy of a seed-based connectivity map, it's literally a seed-based connectivity map. Except that instead of being at the voxel level, it's been compressed or embedded in the space of my parcellation. So now within a parcel, I have the exact same values everywhere. So obviously I've lost some details compared to a seed-based analysis, but the beauty is that I can summarize all possible seed-based parcellation map with that matrix, which is only 1000 to 1000. If I were to do that at the voxel level, well, my God, I would have like 60,000, like 60,000. That's gigantic. To give you an idea, a megapixel is 1000 by 1000. So 60,000 by 60,000 is what? 3,600 megapixel. If you know about a 3600 megapixel camera, let me know, because I believe that would be a world record. So, you know, if we were to do that at the voxel-based resolution, that would be massive. But with the parcel, we're able to do it. And something else that's neat with that representation is that we can see the network. There's this like little square on the diagonal. And that's what a network is. So before we get into that, let's see how we can build this connectome using code. So Nylearn has this a series of tools to build connectivity matrices. So we're going to import it. So from Nylearn.connectome, we're importing that thing called connectivity measure, and we're going to get that object with the kind of measure we want is a simple correlation. And then we're going to fit and transform a data set. And that data set are going to be those time series we had extracted from the parcels. So the 444 time series. Now there's a little bit of NumPy like put in, we say in Quebec here because. And so we can then plot that matrix here. And that's what it looks like. And as you see, it has these like fairly neat squares on the diagonal, just like I had told you before. Now, those squares on the diagonal, they're not going to show up by themselves. You need to order the parcels in a smart way so that you see the squares on the diagonal. And here I've used an option in Nylearn, which is a reordering. And that option was called average. And we're going to talk like in a short while about what that means and what actually that function does, that option does. But for now, suffice to say, here's a connectome, it's ordered correctly, and we see those neat little squares on the diagonal.
A couple of quick questions about that.
Yeah.
First, I know the signal in the white matter is often disregarded for multiple reasons, but are there parcellation strategies that try to do simultaneous parcellations of white matter and gray matter?
Well, you could argue that ICA is a parcellation approach. I mean, the last section of the talk, I don't know if I have time to do it, but that's about ICA. And it's typically run on the entire brain, including the white matter. There's no neat looking, you know, cognitive parcels that emerge from the white matter that way. There are people who think there is interesting functional signal in the white matter you can capture with fMRI. I believe that signal is, if it exists, is weak. And I've never seen, you know, neat white matter parcellation emerge from fMRI, at least using ICA, or you see mostly noise in there. But maybe it's feasible, and but I'm not aware about that.
Great, thanks. And second question, actually kind of related a little bit. You touched on ICA. Would you rather use the mean signal within a node of your parcellation or the first eigenvector or some other metric that would better explain the signal?
The first eigenvector and the mean signal are extremely highly correlated. So I think this is a controversy from the 90s that's still present to this day. And I think in practice, it doesn't have a big effect. But in the long list of parameters, you may want to, you know, swap in a connectome pipeline. That's one of them. And some people are going to say, you know, oh, this was, you know, 1% more accuracy, so it's better. Here we go.
Thanks.
So I was telling you, oh yeah, the column in our connectome, they're like a compressed connectivity map. Well, let's visualize that. So we already seen this inverse transform method in the masker. We've used it actually to go back from a seed-based connectivity map at the voxel level back to a brain map at the voxel level. But those methods also exist for parcellation. So if you have a parcellation masker, which is the case with the first masker we've created, if we apply an inverse transform, it's going to take values in the parcellation space and bring them back as a brain map. And each row or column of our connectome is a connectivity map. So here I'm slicing it through a row, but I could have done a column as well. The matrix is symmetric, so it's equivalent. And yeah, there's a little bit of reshaping going on here, like for obscure reasons. It's like I don't want to spend time commenting on that, but it works, and you get the map. And then we can plot it. So what I've done is plot simultaneously just one sagittal slice of the map that's generated from the connectome and the map that's generated from a seed-based connectivity analysis using for the seed the same region, right? Like here I've used the parcel number 316, and that's also what I had used to extract my signal to do the seed-based connectivity map. And so as you can see, they kind of look the same. You know, you've got the blob in the posterior cingulate cortex, the blob in the medial frontal cortex, I mean, extending to some degree to the anterior cingulate cortex as well. So, I mean, qualitatively, there's a match. You can also see that there are some differences. I mean, obviously, there's a compression, and the blob kind of extend a little bit more with the parcellation-based approach. So we've somewhat degraded our map by compressing, but considering we went from 60,000 voxels down to 444 values, maybe that's a price to pay we're pretty comfortable with. So that reproduces what we had seen in the slide originally. And so now that we know how to build all the elements of this, we can finally get to the parcellation itself and those squares on the diagonal. So here I've highlighted one of those squares on the diagonal. And actually, in that particular example, that square is a default mode network. So you can see it here in all of its glory. It's now just a parcel, like it's either 0 or 1. You are in the default mode network or you're not in the default mode network. And what the red square is just an outline of all the connectivity between little regions within that network. And the reason why it makes a square is because I've ordered my matrix in such a way that all the regions of the default mode are next to one another here. So if I look at all the connectivity between regions of the default mode and regions of the default mode, so it's going to end up living in that little diagonal square. And so why do I call it a network? Well, the basic sort of definition that most people use is simply that the connectivity within that group of regions is larger than the connectivity between those regions and other regions in the brain. So it's somewhat integrated connectivity within and segregated from the rest of brain parcels. And that would be the definition for a functional network, or at least the most standard one that people would use in functional MRI. So let's see how we can do all of that now. Ordering the region, finding the squares, showing the parcels. I'm not advising actually you follow this code necessarily to do it in real life. There are much better instructions to do that very easily with Nylearn. But I'm going to show you all the steps so you understand what's going on under the hood with a little bit more granularity. So the first thing we're going to do is a hierarchical cluster analysis. And that's one of the oldest type of cluster analysis there is. Actually, I think Aristotle did this like classification of nature, or he had something like that, I think, in one of his philosophy books, where he was doing kind of a classification problem. Here, I think, I mean, this particular brand of clustering from the 70s, and it's implemented in the SciPy library, so a very general purpose scientific Python library. So there's a clustering module. Within that clustering module, there's a hierarchy module, and from them we're going to import 3 tools: dendrogram, linkage, and cut tree. And in that slide, what we're looking at is a linkage. So linkage is actually running the clustering. What it's using as an input is our connectivity matrix. So it's going to put together regions that have similar connectivity profiles. And basically what it does, and it's represented actually at the bottom here as a little tree, is that like at the bottom, what you have is like all the parcels in the brain. And those parcels are going to be merged together. And it's going to examine, okay, what are the parcels that are the most tightly connected? All right, those two parcels are merging. And now it's a cluster. And I'm going to do that again and again and again. And at some point, you know, I need to be able to compare two clusters. Before I, you know, at the beginning, I'm comparing two parcels, but then I need to compare two clusters. And what that method average is doing is that it's going to say, okay, so the connectivity between two clusters of regions is simply the average connectivity between those pairs of regions. And that's it. Then you kind of iterate. Now here's a sort of similarity matrix, actually the Euclidean distance between the connectivity maps. One of the metric you can decide to use. And so what the procedure gets you is not really a parcellation, but really like a hierarchy of parcellation. And that's represented here as a tree. So here you have all the series of mergings that are being represented. And at the very end, you only have like two big clusters, actually one gigantic and one tiny, which are going to be merged into one big cluster, which is just all of the brain. This type of analysis representation, it's called a dendrogram from the Greek dendros, which means tree, which is also the same dendros you have in dendrites, which is the end of the tree of the neurons. So that's it. That's just a visualization of that clustering process.
A couple of questions. And there's periodically there's questions about they're specific to NILearn, so I'm not going to bring those up just because we can either take them at the end or address them. So I want to keep the focus on sort of the conceptual stuff. There's a question. Can you elaborate on the appropriate order of parcels within the connectivity matrix? And I think the point here is that it doesn't generally matter except for visualization, right? For most algorithms, at least.
Yeah, absolutely. The order is mostly to get the squares of the diagonal. As an aside, there's always the topics of visualizing high-dimensional objects in low dimension. And there's a bunch of techniques like principal component analysis or, you know, t-SNE or more recent variant of that. But visualizing a matrix of correlation or of distance is a way to understand the topology of your object in a high dimension. And reordering the matrix lets you understand that structure. And so, you know, in a way, it's a very powerful tool to understand how multidimensional objects are organized without having to project them into a low-dimensional space. So it's a little bit of an obscure subfield of visualization, but it's one I'm very fond of and I use all the time. So that's why I really wanted to introduce that in the talk, because that's how I understand parcellation. And I don't have example, but you have much funkier structures and clusters. Cluster has just square the diagonal, but you can have tubes, you can have cycles, and you can learn how to recognize those topography in Connectome, basically. And the ordering is a key ingredient to make the structure apparent. So now that we've got our hierarchy, we've done the dendrogram analysis. So dendrogram analysis, it's going to give us the order. So basically, to be able to make that neat tree, so dendrogram needs to reorder the leaves of the trees, which are our brain regions, such that you know, it merges things that are next to one another. Otherwise, you know, the tree would go crazy and there would be lots of lines that cut each other. So by construction, when you represent a hierarchy as a tree, you order your leaves in a way that respects the clustering structure. And so you can get that order actually from the output of the dendrogram function. And what it's going to look like is that it's a vector of length 444. And it's going to tell you where to put each region. So the first one you have in the vector is the number 372. So that means that when you order your matrix, you should start by the parcel number 372, and then 385, and then 4, and then 53, and so on and so forth. So that's what this vector means. So here we go. Now we're going to look at the clusters themselves, the parcellation itself. And for that, we have to cut the tree, right? Because the hierarchy gives us a series of parcellation. At the very beginning, the parcellation is like every parcel would be one brain region. At the end of the hierarchy, all the brain regions are into one giant big parcel. And whenever we stop in our merging process, it's going to give us a parcellation in nk cluster, let's say. So here we're going to say we want 17 clusters. And why 17? Well, we'll talk briefly about that, but basically whatever would work. 17 is a good number. Let's try that. And what it gives us is now a parcellation. And for that kind of parcellation, what it is just some labels, integer labels. So here I have 17 clusters, so I'm going to have 17 labels because Python loves zero ordering. The labels are from 0 to 16. And so if I look at the beginning of this vector, which is going to be of length 444, because it tells me for each region of the brain in what cluster to put them. The first value is 0, so that means my first parcel goes in the first cluster. Then 1, then 2, then 3, then 4, then 5, and then 5 again. So that means that parcel number 6, 1, 2, 3, 4, 5, 6, and 7 here go in the same cluster, and that cluster is number 5. So if I would look at the parcel number 10, which would be 8, 9, 10, it goes in cluster number 8, together actually with parcel number 11. All right, so I hope this clarifies a little bit what all that data is stored. But if I take that partition here, do a little bit of mixing to put it in the right format, switching to one indexing, so going from parcel 1 to 17 instead of 0 to 16, and I apply an inverse transform, I can get back a brain map, which looks just like what we've been discussing from the beginning. And that's exactly, when I've spent at length like trying to explain you more from like a linear algebra perspective what like those integer indices mean, but they're just a color in my map, right? Like each color is an integer number. So, I've colored my clusters using a given color, which happen to be in NumPy, a given integer. So here we go, we have a parcellation. So that's the key things I wanted to cover today. Now we're going to see a bunch of other points, but I think like this is a central one. That's how you build a parcellation in fMRI. And it looks like we have a default mode network, by the way. It's in orange. It's great. So just as a quick aside, parcellation can also be represented using what's called an adjacency matrix. And what an adjacency matrix is simply a matrix where you have the regions times the regions, so like the connectome. But instead of having the connectivity measure in the matrix, what you have is 0 or 1. You have 0 if the two regions are not in the same cluster, and you have 1 if they are in the same cluster. And what I've done here is that I've created a little function that converts a partition, so that vector with integer labels, into an adjacency matrix. And if you're curious, you can see how that was done with NumPy. A little bit of a trick with broadcasting and transposition. Repetition of vectors and transposition. But without getting into the details of how that procedure works, what I've done is that I've reordered the partition based on the order of the hierarchical clustering. And that's how you do it. Because order is just this vector of integer. It's possible to just like index your vector with the order vector. It's going to reorder it. It's a bit confusing why that works, but it does. And I do the same with the connectivity matrix. So here I reorder the rows and then I reorder the columns, all right? And I get back my connectome with the nice squares of the diagonal. And actually that's exactly what nylern does to give you that. So now we've understood what nylern, the plotting matrix function of nylern does. And if I reorder my adjacency matrix, boom, squares on the diagonal. So when I was telling you a cluster, they're squares on diagonal, it's a literal thing. Like if you take your adjacency matrix, which encodes for the partition, and you reorder it with the order from the hierarchical clustering, wherever you're going to cut your hierarchy, you're going to get those squares. And actually, if you were to visualize the hierarchy at different scale, at different numbers of cluster, you would see those squares merge with one another and become progressively bigger. All right, trying to capture, small squares in the connectome and then big squares in the connectome. So I just wanted to go back real quick to the compressed map, which is I've shown here again, and put next to it the DMN cluster we found with our hierarchical cluster analysis. And as you can see, it matches pretty well. Although the actual square that the procedure finds may not match exactly what a thresholded connectivity map would give you, it sort of matches our intuition that basically the connectivity map has found the network. And that if we were to just like somewhat threshold, maybe with correcting for multiple comparison this map, we would find the entire network. Well, that intuition is not really off. But the clustering process is a really multivariate process where it looks at all the regions together to decide how to define the square, rather than focus on a particular seed, which in that case was the posterior cingulate cortex.
Here, you might get to this. This is sort of the big question, always comes up. Is the way to check how many clusters you should choose to best characterize the data?
This is indeed a very common question. Every time I present that, and every time I submit a paper, a reviewer will bring that up. I have an entire section on this, so just be patient. I'm going to just check on the time. So it's been 50 minutes. I'm actually not doing that bad. We can go for another 40, right, Tal?
You could, I mean, in theory, you can go for another hour and 10. It's nice if you leave participants some time at the end, but anytime if you want to take a break, we can do that anytime.
No, 2 hours too long. I think we're actually, it's going to be an hour and a half. So if there's questions, we can also come back at the end. All right, so as I was saying, really like what we've covered so far, it's like what clustering is. I've taken the example of functional MRI, but really most of what I say would apply to pretty much any data array. I would say that I've presented it through the sort of lenses of a connectome and of reordering a connectome, and that maybe not that standard. But basically, functional parcellation in fMRI is kind of based on functional connectivity. It's kind of a fancy name to say we're doing hierarchical clustering on the correlation matrix. So that was kind of like the key point I wanted to cover. Now, obviously, there's lots more going on in the literature about functional parcellation. So the second half of the presentation, what I'm trying to do is cover what I think about as like hot topics, and selecting the number of clusters is one of them. Some of them of those topics are still quite active research-wise, despite functional parcellation being something like very active, I'd say, for 20 years now. There's still some work to do. So one of them is how do you do group parcels, and how do you go from individual to group parcels? Because what I've shown you here was for an individual analysis. There's kind of like 3 big families of techniques. And I would say to some degree, it's kind of like, the oldest and then not as old and the most recent. So in somewhat of like older approaches, what you would do is simply build individual parcellation. And then you get your individual parcels, and somehow you're going to combine them to give you a group parcel. That would be one way of doing it. Then people started thinking, we get those patterns as a group level, but could we use that as kind of a prior for the individual level? Because, there's a noise at the individual level. So maybe we're going to miss a network, or maybe, there's many ways of doing a good parcellation for an individual. So could we enforce to do the same kind of parcellation as what we converge to at the group level? So in that like middle schematic, you do your individual analysis, then you recombine them, you get a group level result, and then you're going to go back to your individual level, but now using the group result as a prior to constrain the decomposition you're extracting. And possibly you could imagine somewhat like looping on that. And then you would get what you have on the right, where really the individual and group level analysis become somewhat connected and actually can be done in a joint approach. So there was this very nice paper from Liu and colleague in Neuroimaging in 2014. There is a more recent paper, which I forgot to add to that slide, by Thomas Yeo's group. And unfortunately, the name of the first author escapes me. I'm wondering if it's Kong et al. Maybe. So anyway, there's a recent work that still tries to do this right, like finding models. So you typically using Markov chains and Bayesian analysis, multi-level, to try to combine individual level and group level analysis into one single estimation. So that's that. There's lots of different technical solutions out there, and a lot of groups kind of stick to their guns, like there's the techniques they're used to, and that's what they do, also based on whatever software package they're using. So even currently in the literature, you're going to find like a vast sort of spectrum of practices in terms of how people infer group and individual parcellations. So let's do a group analysis. We're not going to get scared. We could actually do some group analysis directly with some nailearn objects. We're going to do that at the very end. But let's do one from scratch just to get a feel of what it would look like. Actually, that group analysis is not very far from what I proposed in 2010 with Basque algorithm. It's a tad bit simpler though, but in spirit, it's not too far. What we do is that we're going to create an array of time series array. So we're going to initialize this time series all empty array, and then we're going to loop into all the data set we fetched from ADHD. We had 10 subjects that we fetched. And we're going to just append that to our array, all right? So the first element of this array will be a 2D array with a time series of subject number 0, and so on and so forth, up until subject 9, which is the 10th subject of our analysis. I hate zero indexing, by the way. I find this so confusing. But, the 10th subject, number 9. Then what we're going to do is that we are going to compute our average connectome. And that we can do in a one-liner, because that connectivity measure tool we've already used from nailearn, it actually computes the average connectome too. So all we have to do is fit our array of arrays, and that tool actually understands that. It expects that, really. That's why there was a little bit of like manipulation in my previous code. It was to actually create an array of array, whatever. And it's going to give me that object, con_all, which has all my connectomes along with also the mean connectome. And it can be accessed through that attribute, mean underscore. So I believe that's already been said, but in nailearn, attributes with an underscore they're things that have been fitted from the data, right? So that's your mean connectome. Then we're going to apply, now that we have the mean connectome, we're just going to apply a linkage on it. So basically, that group level technique is the same as individual level technique, except that instead of working on individual connectome, we're working on an average of individual connectome. So that's that. So I get my hierarchy. I'm going to cut it just like I did before. And then I'm going to take my partition and that I'm going to inverse transform it to get a brain mass instead of just a vector of integers. And I'm going to plot it with a dedicated plotting tool, which is Plot3, which we've already talked about and seen a couple of times. And that gives me those parcels here. They're not particularly nice. I'm actually missing the default mode now. Whatever, it's only 10 subjects. So, but at least you've seen one example of a simple group analysis. All right, so now we come to multi-resolution parcellations. So that's a question of like how many clusters. So before we talk about those different resolutions and what happens when you change the number of clusters, I'd like to point out that there's actually two families of clustering techniques, and that what we've covered is the one at the bottom. And this family proceeds by looking at small regions that have been pre-computed somehow. And then you can build a connectome and do expensive clustering techniques such as the hierarchical clustering. But that only works because that matrix is not too big. It's only a few hundred columns and rows. We can deal with that, no problem. But you can also try to do that at the voxel level, but typically you won't be able to build that matrix, and you're going to need to use techniques which don't require that. And so k-means can operate at the voxel level because k-means doesn't store like all voxel to voxel similarity. It's only looking at voxel to cluster centroid similarity. You can do hierarchical clustering, but under a spatial constraint. So instead of looking at the most similar regions across the full brain, for each region you look at the most similar spatial neighbors, which typically you have 6 or 8 or 26 of them. So it's not too big. And actually this type of region growing approaches can give you the small regions to then move on to a full brain, spatially unconstrained approach. So based on those different techniques, you're going to get very different resolutions, right? Like you can have very large scale network where you have few clusters that are distributed throughout the whole brain. And typically what I just presented you did just that. Or you can have small cluster, high resolution, lots of small cluster, which is high resolution. And that would be the type of technique at the top. And basically what emerged very clearly from decades of research is that you can get meaningful and useful parcellation at all those different resolutions. So very much in line with actually what Varela was presenting, that you have all the series of like multi-scale functional assemblies. You have multi-scale functional parcels. So let's go through a zoo of multi-scale functional parcellation. This one is one of my all-time favorite. I really like it. It's got 2 parcels. It was reported by Yuva Golan and colleagues in 2008. And what they found is essentially all the sort of like sensory motor, visual, auditory cortices on one end and everything else on the other end. And what this everything else would be is, posterior cingulate, the frontal cortex at large, and part of the temporal cortex. And what they mentioned is that, maybe those are more like exogenous parts of the brain, driven by what's outside of us, and endogenous parts of the brain, which are more concerned into, dreaming, planning, working memory, things like that. Interestingly, there's one of the first paper I know in functional brain parcellation is by Luria, who's a famous Russian neurologist. And he was proposing there was 3 networks in the brain. The frontal cortex, everything else in the frontal cortex, and then the sort of points for everything that's like autonomous function. So if you, based on Luria, for cortical, he said there's a frontal cortex and all the rest. And, you know, close enough. He missed the back of the default mode network, but that's more or less what is here. So that results as roots far in the past. It's also very current because right now there's a lot of buzz around the notion of gradient of brain function. And what the primary gradient is actually a gradient between those two networks. So people are you know, kind of rediscovering this mode of organization and commenting it extensively through gradient analysis techniques. So that's a way of parcellating the brain. Then there's the iconic Euclidean parcellation 7 cluster. It is in nylon, so here I use plot1 to visualize it. I've also used a view image to visualize it interactively, and I saved the frame so we can look at it together. So here we go. You've got your default mode network. Very nice, actually fits very well with what we've done, except that here you also have the temporal components, which on the individual subject, we didn't have it. Oh, my laptop is about to die. That is not good. Okay. Then you have a sensory motor network in green, which was the first one discovered by Bisval. I mentioned that work. Then in blue, you have the visual network, very clean. And then you have all those weird frontoparietal networks. In red, you're going to have the dorsal attentional. Then, so it's got like the frontal eye fields and all a bunch of regions here. And for me, I call it the Pac-Man network because you have the two eyes and you have the, you know, the smile here, or the smiley face network. So anyway, then you have the ventral attentional network, but really it has a bunch of regions that are in the salience network too, based on other works. So the terminology is a little bit like not all over the place, but at least not completely standardized. And then you have what Yeo calls the Yeocrinian calls the frontoparietal network, or executive function network here. The last one of the 7 we haven't talked about is the mesolimbic network, which has the temporal pole and the orbitofrontal cortex. And interestingly, in the Yeo paper, that network is not commented on much, and I believe it's because of the low SNR, signal-to-noise ratio, and I believe Yeo decided to just exclude it on that basis. Interestingly, I had a paper about the same time where I also decided to exclude it for the same reason. I'm going to change room to try to have my computer not totally break on me. Hopefully, I will not lose the Wi-Fi in the process of doing that.
You want to give us a tour of the house while you're walking through there?
I'd rather not. My house is a total chaos. With my kids, I should have got a charger prior to do that, but you probably don't want to really see how my house is. All right, so a little bit of adventure. So yeah, all right. So let me just get out of food. Yeah, I'm charging. Everything's good. So yeah, so the mesolimbic network, I actually, based on more recent work, think that it's a legit network, that it does carry some information actually in some clinical conditions. But it remains a kind of like controversial network that not everybody includes in their list of standard functional networks. But the reason why I sort of guided you through those is that they're really like classic and the names that Hio came up with now are used in a bunch of papers, and actually those particular networks are used in a bunch of papers. But now already with in that very same work, they had noticed that if you ask for 17 clusters, you would also get some fairly good and interesting results. So that's the slide after, and I also have a little interactive viewer here. So one of the things that they comment extensively in this like massive piece of work, I think the paper is like 70 pages long, is that the visual network somewhat subdivides itself into like this dark blue, light blue, and the green areas, which actually go and connect with sort of like parietal cortices. And you know, they discuss that this likely reflects you know, both some visual area and also the extrinsicity of the visual field may also drive some of those parcellation results. So it looks biologically plausible, and I think, I mean, there's lots of evidence by now that you can get some fairly fine functional parcellation that still capture you know, interesting and biologically meaningful features. So it's not just big networks we can capture with this type of analysis. We can also get subnetworks and possibly go all the way to the regions themselves. And yeah, so that I'm going to close now. And so there are now some atlases that offer a series of scales, a series of numbers of clusters. My group has developed one, which is called the BASC. It's a MIST atlas developed by Sebastian Erks. The most popular one is a Schaefer atlas, which is also from the Thomas lab, and also is in Nylern. Here I've done this little like loop through the BASC entries. So we were looking at the 444 parcel solution, but here I've done a little loop where I plot the BASC atlas just on one slice, one sagittal slice, and I'm using some subplot from Matplotlib. So, you know, we see them on 2 rows. And you can see at scale 7 in that atlas, which was also the number selected by Yeo, you get those big distributed network, and then they start breaking down and breaking down more. And at the end of the day, you get those things that are much more like region. And actually, most of them are region, like they're connected in space. If you're curious about understanding this multi-scale organization, there's a fairly nifty dashboard that Sebastian's developed, where you can select a big network. So let's look at the default mode network, for example, and you're going to get at the root the big network, and then how it's subdivided here in 3 parts, which are, you know, the posterior and lateral and anterior. So those 3. And then if you're interested in, say, this one, you can always click on it. And then you're going to see that particular guy subdivides further and further, and so on and so forth, all the way to looking at tiny little regions. So it's a neat little tool to try to explore the hierarchical organization of those parcellation if you're curious about that. So that's that. Why can't I? Okay. And in the 15 minutes that's left, we're going to talk a little bit about quality of the parcels. So we just covered the numbers, and the takeaway message is that there's multiple numbers that are viable. There are some techniques to select, you know, the best number or the best numbers, plural. I believe they're quite noisy, and they're kind of like, I'm not sure they actually converge to any number that's more or less meaningful than another one. I think the right way of thinking about number of parcel is a little bit like if you were to compress a natural image. You know, you can do what's called color quantization, where you approximate a whole range of color into one. And, you know, if you have only 2 colors, you have only a very crude image. And if you have 10 colors, you have better image. And if you have 100 colors, you can see like a lot of details. So, you know, depending on whatever amount of detail you think is good for analysis, you may change your number of clusters. But there's not a magic number that's going to tell you what the right number of cluster is. And I headbutt with reviewers again and again and again on that. And I will continue doing so because I just don't believe that there is a number of cluster that's more meaningful than another one. So maybe slightly controversial take, but that's a strong opinion of mine. So are there any question on the more like multi-resolution aspects?
I was about to say there are none, but the one just popped up. Oh, it's just, can you post that link? I'll do that, that's fine. Otherwise, no questions, but feel free to ask them, folks.
All right. So now, how do you determine if a parcellation is good? Because, you know, I showed you all those different parcels, all those different algorithms, but what is a good parcellation? And one of the criteria that people have used is the homogeneity of the cluster. So whether the time series within a given parcel are highly correlated with one another. That would be the simpler sort of definition of it. In general, you can look at the compression quality. So if you compress a brain map in the parcellation space, does it resemble the original brain map? And that definition of compression applies both to traditional parcellation, like what I've just shown you, or more like decomposition-based parcellation, like ICA, which I'm going to talk about in a few minutes. So in terms of homogeneity, I love this graph. It's from the Erks paper for the MIST Atlas. And what it shows is the homogeneity of different parcellations as a function of the average size of the parcels. And the correlation between those two things is like 0.9 something, all right? So here you have your Yeo 7 atlas, your Yeo 17 atlas, the Glasser atlas is here. In blue, you have the Basque multiscale atlas I was presenting, which Sebastian called MIST, but those are actually literally the same atlases. Then you have the Glasser atlas, which is a very popular atlas from the Human Connectome project, the Brandnetome atlas from China, the Gordon atlas from Peterson's group, and in red you've got random parcellation generated by Cameron Craddock, and they all perfectly align, and it's really sad. So much work to get good parcellation, and really, what matters is how many parcels you have. So my analogy with the image is really like, if you want more details, just use more parcels. The precise algorithm, the precise parcellation you use, actually, in terms of that particular criterion, doesn't seem to have much impact. I mean, I could tell you, oh my God, look at that, the MIST 36, the MIST 20 is about the same as Yeo 17. So, but I mean, those are tiny differences, and much, much smaller than what you would have by just changing the number of parcels you use. So I don't think there is a better atlas in terms of their homogeneity, which leads actually, the choice of atlases today, I feel, is mostly based on convenience. What is it you have access to? Do you like the labels associated with that atlas? And is the number of parcels appropriate for what you're trying to do? But I don't think that people are like, oh yeah, that parcellation sucks, and oh, this one's great. In practice, they behave pretty similarly, at least in terms of homogeneity, clearly. We're going to talk about other criteria where there is some differentiation, but for the homogeneity, there's no clear advantage for particular approaches. So here, I'm not going to comment on the code. That's atrocious code. I've done it fast, and it's very hard to read. But I really wanted to show you the figure. So what I've done here is a little loop where I've redone what we have done before, which is to extract a parcel from Basque and compress the connectivity map, and to show you the compression of the map over different number of clusters. And so what we had looked at before was like 444 against voxel-based, and here I've added you 64 and 20. And as you can see, I mean, 20 is not that bad. You get the blobs at the back and the front. Now the blob extend a lot, and it really looks like this kind of like a stained glass in a cathedral rather than the cover of a neuroimage paper. But whatever, it's only 20 values, so it's very compact. So maybe for some usage, it may be useful. I've actually shown that when you do connectome-wise statistical testing, like running a linear regression model on every edge of a connectome, using 20 clusters is actually a very reasonable way of doing that. You get lots of power, just because you reduce the number of multiple comparisons you have to deal with. But obviously, if you want to get a good approximation of the map, you would rather go with 400 plus here. So that relates to that question of homogeneity. The more the parcels are homogeneous, the better they approximate the original signal, and the better you're going to approximate the map at the end of the day. So another important topic is how reproducible spatially those parcels are. That would be a criteria to get a good parcellation. And for the group reproducibility, I should say that at least if you have an homogeneous population and several hundred of subjects, this converges toward very stable results. And actually in the Yeo-Crinan paper, they had created a discovery and replication sample with 500 subjects for each parcellation. And the level of reproducibility is very high. Now, if you vary things like, a template generated from group A against group B, and group A and group B have very different characteristics, maybe it's not going to be as high. If you start, changing the algorithms, then you can introduce systematic differences. So it's not like there is one group atlas to rule them all. Different technique will lead you to different group atlases solution. But at least, within an algorithm and within a population, group atlases can be made really reproducible. Now, at the individual level, it's much more challenging. There's this beautiful paper a few years back, the Midnight Brain Scan project, where 10 individuals got scanned every day for a few weeks. And so they had 10 runs of 30 minutes of resting state, so 5 hours total. And with that amount of data, they were able to show a beautiful individual parcellation and show that there is some specificity in the parcel, like aspects of the parcel you will only found in some individual and not at the group level. So there's this idea that the parcels are really specific of each individual. You cannot really neglect, because you've co-registered your images, just say, oh, basically the group parcels are just going to work for the individual level. So there's now pretty solid evidence that there is a need to adapt the parcellation to each individual. And as I said, how to do that is still a subject of active research. But in terms of how much data you need to get good individual parcels, there's this neat graph in the Gordon paper with a dice overlap between sort of different data sets, splitting the individual data into subset and seeing to what degree the parcellation reproduce with the dice index. And there is definite gains when you go to about 20 minutes, which is substantially more than most, at least, legacy study collected. And even to this day, clinical studies typically more collect between 5 and 10 minutes than 20 minutes. But what I find interesting here is that you've got this plateau that even when you go to over an hour of data, you see very marginal improvement in the reproducibility. And that's a bit of a problem. You don't get perfectly reproducible individual parcel. And my current thinking is that this is due to neglecting the dynamics, that there's actually states of parcellation at the individual level, and that trying to find one parcellation that fits all your time series. You're trying to average some things intrinsically heterogeneous. So the mean is possibly not meaningful. And in any case, it will take a very long time before that mean sort of like stabilizes. So that's from the Gordon paper, but that's something I've reproduced in the lab. And there's also very deep down into the supplementary material of the latest paper from the Milans group with Aki Neocolaides as the first author. They have the same result. So I think this is a sort of not just marginal observations. There's really a challenge in terms of finding good individual parcellations. So I've put you a little bit of code here to show you how to compare parcellations using the DICE index. But because I'm running a bit out of time, I'm going to skip that. Basically, scikit-learn has already implemented a number of metrics to compare the parcels. The most standard one is this adjusted rand index. I've put a few comments to explain to you what it computes. It's related to the adjacency matrix that we've seen together. It's basically comparing those two adjacency matrices and looking at the proportions of elements that are the same, and normalizing that by sort of like a chance level value. So that's what a normalized rand index is. And so that would be one way of quantifying the reproducibility. Just as a funny sort of comment is that, like, here I've compared two individual parcels from two different subjects of ADHD, and the adjusted rand index was 0.1, which is very weak. You know, it behaves a little bit like a correlation, right? Like a good value would be close to 1. So 0.1 is very, very low. But, you know, here we have very short time series. There is a lot of individual variability. Also, we've used a very like crude algorithm, just this like average linkage. So I've added, so that's an hyperlink, like you can click on this, to the paper of Nicolaides I was mentioning earlier, where he benchmarked a bunch of algorithm and duration of time, etc. Both for group and individual analysis. So if you want to learn more about what are the critical parameters, that would be a good paper to dig more into this. And finally, the linear decomposition models. So far I've only talked about what's called hard parcellations. So where you are in a parcel or you're not in a parcel, no room for compromise. But there are many other types of decomposition methods that have been proposed, you know, since a long time ago, and applied to fMRI also for a long time. And those decomposition methods, instead of having, you know, the type of integer labels that I've been presenting all the time, they are based on a linear mixture model. Very similar to the sort of linear regression model you use when you do activation-based fMRI. So essentially, you decompose your 4D data set into a series of brain maps, which would play the role of your clusters. So those are your parcels. And each brain map is associated with a time course. And in some way, you want the linear mixture, like if you take all of those time course, for each voxel, you associate them with a weight that's based on those maps, and then you add them. You want this linear mixture to somewhat approximate the original time series. Now, there's lots of different criteria. You can ask those maps to explain as much of variance as possible in the original data, in which case you do a principal component analysis. You can ask also those maps to be spatially independent, in which case you do spatial independent component analysis. Then you can do a PCA, but ask for your maps to be positive and also sparse, have only a few voxels that are non-zero. And then you do, you know, a sparse positive PCA. So there's lots of variants, but what's important to understand is that those relax some of the key hypotheses we do with traditional clustering. Here, our parcels can be smooth, so they can be kind of gradients. Also, our parcels can only really contribute in some points in time. So here, this type of method can capture some of time-varying parcellation, which a cluster analysis can't really. Like, it's looking at the correlation structure by aggregating all available time points. So, but some things can be also confusing. Like for ICA and PCA, you can have negative values in your maps. So, the negative value of one map can compensate the positive value of another map. So it's somewhat hard to assess exactly, what's the contribution for a particular voxel by just looking at the map and the time series of that map. You really need to look at all the components jointly if you want to really understand what's going on in your data based on that summary.
Pierre,
So
just
yeah.
before you go on, there's a question that you touched on a little bit. Has anyone tried to look into how individual parcellation might change across time?
Yeah, so there's a paper from my group that looked precisely at that. It should be published soon, but the preprint is out there. Amal Boukti is the first author, and I'll share the link. And yeah, they do very strongly. So I've tried to put in there things that I feel are not too controversial. I think that the most controversial part I mentioned is the number of clusters and the fact that it's a waste of time to try to find the best number of clusters. That may be a bit controversial. But yeah, the stuff about the dynamic of parcellation is, I feel like, still something that's current and being debated, and so it's not in that presentation for that reason. So yeah, the decomposition. So NiLearn has some decomposition method, a bunch of them. ICA, dictionary learning, so with sparsity, which I talked about. Also, that slide is the first time, like, that's code you could actually reuse. Like, the other code was more to try to show to you under the hood how things are done. That's a real analysis with NiLearn, using the NiLearn sort of syntax. So from the NiLearn decomposition, we're importing a dictionary learning method. And the dictionary learning has an initialization, which we are doing here. So we're going to ask for 17 components, staying true to our 17 clusters. And all the options we had before in the masker, they're right away part of that method. So that method is going to work directly from the 4D data, the Nifty files, and do the masking as part of the analysis. So we are detrending, we are not standardizing here. Dictionary learning method typically use the variance information. So we are not going to correct for variance here. We apply a bit of smoothing, and those are not like very important options. And then we're going to fit our first subject, just like we did in all the other examples. And then instead of plotting of using plot ROI, which sort of assumes that you have one 3D volume with labels in it, we're going to use plot prob atlas, probabilistic atlas, because here we have 4D data. Like, each parcel is its own volume, because there is possibly some overlap between your parcels, so you cannot stick them all in a 3D volume. So you have a series of different maps. And what prob atlas does is that it sort of thresholds your maps, draws an outline, and puts all those outline on a single map. And it doesn't look great in that example, but it's actually, some of those components are really nice. It's just they're a bit, because there's very little data, there's some point in the brain which end up not being in any parcel, which is something that can happen in the decomposition model and will not happen in the clustering technique by sort of definition. The cluster are going to cover the whole brain, but decomposition doesn't necessarily does. And here I've sort of manually selected one of the components, and it says daemon component, but it's not the daemon component, because I think there's some random aspect of the tutorial, and I rerun it. So my number is not the correct one. So you're gonna have to trust me. In there is a component that looks really like the DMN, and that's actually not too far from our hard parcel that we had extracted. So to some degree, there's some convergence between all of these techniques, and that was, I wanted to show you an example where there is a nice convergence. I was killed by my random seed, but I'm not sure why, because I actually did control for the random state. It's there, but whatever. There are also some differences, qualitative differences. There are some, the ICF, for example, finds lateralized attentional network very consistently, which the clustering typically doesn't, or at least not at low resolution. But it's not completely clear to me what causes those divergences, but there are some. So here are other example of how can you judge that a parcellation is a good parcellation. So we talked about reproducibility, we talked about homogeneity. And here is a different approach, which I think is really interesting. So it's a beautiful paper by Daddy and colleagues in 2020, but it's at the archive I link, but it's in your image. And they tried to do some classification task using different atlases. They've done a bunch. I'm just showing two here, schizophrenia versus control and IQ versus low IQ. And what I find interesting is that there's very little differences between the different parcels. On the x-axis, you have the resolution, the number of parcels. And you can see that both the resolution and the parcellation being used have only marginal impact. So for example, in blue here, you have an ICA generated from the UK Biobank. In pink, you have the Diffumo Atlas, which is a dictionary learning methods. It's actually the best performer in that benchmark. The Basque Atlas we were discussing is in yellow. You have the Schaffer Atlas in orange here. And I mean, they're all like pretty tightly live together. Now, you can gain 5–10% of accuracy, which is, nothing to sneeze at. And this paper recommends using Diffumo. But, it's not a huge impact. But that's a kind of benchmark that could be used to sort of drive your choice of parcellation. Another interesting metric that they did was the compression, quality of the compression. So that relates to the homogeneity, as I explained before. And here they found a really big difference. And basically the decomposition-based methods, such as ICA or dictionary learning, compress better the maps. So here you have an example of a statistical activation map at the original voxel level, compressed with a Diffumo Atlas and compressed with hard parcels. And you can see that Diffumo looks much more like a voxel-based analysis. And I think it's just like the parcels are much richer, right? Like you have they can have low and high coefficients. They can have negative, positive. So you have much more degrees of freedom to compress your data. But at the end of the day, you use the same number of coefficients, right? Like if you have 100 Diffumo components, your data is compressed in dimension 100. And if you care about approximating what's going on at the voxel level, there I think there's a clear advantage to going with the decomposition-based method. So here we go. We get a summary. There are some networks high within network connectivity and low between network connectivity. Those network or parcels, you can extract them over a range of scales. So from the distributed network all the way to specialized regions. The homogeneity is going to increase when you have high scale. The stability of the parcellation is very high at the group level, but is more difficult to achieve at the individual level. You have now a number of benchmarks to establish the quality of parcels. As I said, they're not necessarily that conclusive in terms of homogeneity and reproducibility. I think in terms of performance in classification, that's more interesting, and the compression has like a clear takeaway message. There are some substantial differences between individual and group. And so, hopefully in the future, we'll have a great method that's very consensual to do those things properly. I don't think we're there yet. And finally, the linear mixture gradient type of analysis give you a richer view of the organization of the parcels, which relate to the fact that they compress them much better And but they're also more complex to interpret. I mentioned quickly this idea of like there's sometimes positive and negative weights, which can be in the map, which can be a bit hard to interpret. So here we go. Here's an image of a brain with frog legs that has been parcellated. And I'd like to thank the Nylon team because I've done like all my code and visualizations thanks to their amazing work. And in the lab, we're now using that for all our papers and such. So I'm very grateful to the community for doing that.
Great, thank you so much for that wonderful talk, Pierre. We have a couple of questions. First, what do you think about creating specific parcellations for each study? For example, when dealing with a diseased cohort.
So I haven't really seen any evidence that could lead to improvements, say, in classification problems like the one I expose here. In terms of the homogeneity, which I have the graph here. Here we go. I think the reason why all those atlases seem to perform a little bit equivalently is that I think all the group atlases suck. As I said, there's a big difference between the individual parcel and the group parcel. And the group parcel will never really fit well anybody. So, you know, you can try to create a group atlas specific of your particular group. So it will suck for your particular group instead of sucking for all the groups, but it will still suck. So, you know, I don't think you're going to see big gains doing that, unfortunately. So that would be my take on it, but that's quite speculative. As I said, I mean, I'm not aware of really work looking at that in great detail.
And last question, unless there are others, feel free to ask. What are your thoughts on the use of multiple parcellation scales simultaneously? So looking at functional connectivity from 10 versus 200.
So I like the idea, it's intriguing. I mean, so the BAS multiscale atlas with Sebastian, we developed it to do that, to be able to do that. So far, what we've done hasn't necessarily led to real insight or, you know, better behavior on the various things we try to do with them. And I've seen few papers only using simultaneously the different scales. But you know, it's a bit early to tell if. I mean, conceptually, it's clearly interesting. In practice, I'm not sure whether it's gonna, you know, bring insights. And hopefully, you know, in the next couple of years, we'll get more paper to get an idea of that.
Great, thanks. And one new question. How about metrics other than correlation coefficients to estimate the level of connectivity? Would they produce different parcellations?
So in the so there's two recent papers that have tried to the DALI paper is one of them that have tried to really look quite systematically at the different choices you can make into a connectome prediction pipeline and how that impacts your final outcome. So that particular paper, I don't know if they looked at the type of connectivity. They had a previous paper in like a conference paper that had this, where they looked at 4 or 5 different, and they found a positive impact of the regularized connectivity estimation with sparsification. So the other paper is by the Oxford group. I cannot remember the list of first author. I hate when that happens because that's the person who did the work, but Steve Smith is the last author. And they also conclude that some kind of partial correlation, regularized partial correlation analysis was better than straight simple correlation. If I remember correctly, and don't take my word for this and check with the actual works, those gains were fairly marginal, but there were gains.
And last question, I'll actually ask a question. I'll push you on your controversial stance, which I completely agree with, but I think it's worth exploring the implications of that, right? So you kind of alluded to this, like, if you want better predictions, generally speaking, and you have the data, you just add features, right? So you would just opt for the highest dimensional clustering or any other kind of decomposition, really, that you can, given the amount of data you have. If you so in that sense, there is no sort of correct answer except like what you can get away with, including voxels if you had enough data. So the question is, okay, so that can't give us like the best in any sense clustering. What does that leave then? So if it's just a matter of does this make sense to me, then are we just going around in circles every time someone proposes a new clustering that looks good to them for this question, looked at it this way under these constraints? Or is there anything we can appeal to beyond just through predictive utility?
Yeah, it sounds like a very nihilistic conclusion. I mean, my response is like entirely speculative. I can tell you like my sort of intuitions about those things is that there is a good parcellation out there, and it's individual, and it's time varying, and it's extremely reproducible, and it leads to very high compression rates. That's something we're working on in the lab, and I believe this will give an edge in classification task if it could be generalized for couplet-level analysis. I think there is tremendous value in having community-based sort of atlases because they are vocabulary for us to exchange. When you're at the voxel level, it's very hard to understand each other. If you have a set of networks that have names that people kind of get an intuition of what they mean, that helps sort of like create an understanding of across studies. So, I think there is a value in parcellation, if only for these more like normative aspects, and that there is still room to improve parcellations. Now, a few years back, I had this almost like fallout of parcellation. I've been doing that for way too long, and I felt, the deep learning is here. You can do convolutional neural nets. You can learn basically how you reduce the dimensionality of your data in a way that's possibly much more powerful than with cluster analysis, what have you. And I was essentially ready to drop parcellation entirely and basically do more like deep learning type of things. There's this new family of deep learning techniques which are called the graph convolutional neural networks that use parcellation, actually can use also multi-resolution parcellation, and are very efficient data-wise, much more so than traditional convolutional neural networks. So that's why I'm still in the game and working on it very actively, is that I think actually those data dimension reduction technique are maybe the way forward to apply deep learning in fMRI without needing tens of thousands, hundreds of thousands of subjects to be able to train a model well. Once again, speculative, maybe in 5 years, I will be proved completely wrong, and deep learning will have taken over with voxel-based analysis. But I feel that combining graph-based representation with deep learning is possibly a very fruitful avenue for the future.
Awesome. Thanks a lot. This was a fantastic talk. I'll clap, but this is on behalf of everyone who's all at home doing the same.
Yeah, it's very lonely, the Zoom presentations.
It really is, yeah. It's hard to know how people receive anything you say.
I hope people liked it, and thanks for inviting me once again.
Yeah. They can tell you they liked it in Slack afterwards, and now people have to, because if they don't, it's like, so don't worry, Pierre. Everyone's going to tell you how much they enjoyed the presentation. But thanks again, and hopefully we'll do this again next year, maybe in person in Seattle. All right, so we'll take a 10-minute break, and then we have 2 more talks coming up. Thanks, Pierre.
Okay. Bye, everyone.